This presentation will focus on the structure and function of myoglobin and hemoglobin. Now, there are 14 globin chains within the human genome. Two of these globin chains encodes a protein that is monomeric. 5 of these genes encodes a protein that has an alpha-like structure, and 6 of these genes encodes a protein that has a beta-like structure. Now, myoglobin is the prototype globin that we'll start off with. Myoglobin is formed from 151 amino acid residues and 1 heme group. The amino acids form ahelical segments labeled A through H, going from the N-terminal to C-terminal ends. For the figure that's rotating on the right, the N-terminal end is colored in blue, and the C-terminal end is colored in orange. Now, amino acid residues can be referred to by their numerical position and by their relative position in the ahelical segments. For example, the 8th amino acid in the F helix is referred to as F8, and the 7th amino acid in the E helix is referred to as E7. Now, equivalent proteins or equivalent globin molecules can be superimposed upon myoglobin such that F8 will always refer to the same amino acid. So therefore, in this particular example, F8 is a histidine, which is covalently linked to the heme group. The heme group, which is shown as a disc on the figure on the left, is wedged between the E helix and the F helix. As I said before, the distal histidine forms a covalent bond to the heme iron, and this is on the proximal side of the heme group. This particular histidine is referred to as the proximal histidine, and it's F8. The ligand, such as molecular oxygen, carbon monoxide, or nitric oxide, binds to the distal or opposite side of the heme group, and that's shown by the letter D. There's another important amino acid, which is E11, which is also a histidine, that interacts with the ligand and forms a hydrogen bond and helps stabilize the bound ligand. In this figure, we show the heme group, which is shown looking at the face of the heme on the left and perpendicular to the heme plane on the right. In this particular figure, the carbon atoms are shown in gray, the nitrogen atoms are shown in blue, and the oxygen atoms are shown in red, and the iron atom is shown in orange. Now, there are 4 pyrrole ring groups, each one with a nitrogen that is coordinated to the heme iron, and the 4 pyrrole groups are connected by methylene bridges to form this general ring-shaped structure. The heme group binds the proximal histidine, which you see in the figure to the right, which is histidine 93 in the numerical numbering, or histidine F8, which is the proximal histidine. In this particular figure, we see carbon monoxide, which is bound on the distal side. This carbon monoxide interacts with histidine 64, which is the distal histidine. Now, as an aside, one of the things that has to happen for carbon monoxide or oxygen binding is it has to get inside, the ligand has to get inside the heme pocket. The generally accepted model is that this distal histidine can swing back and forth, and this allows ligands to enter and exit the heme pocket during the ligand binding reaction. This is really something you don't need to know for the test, but it's kind of a good thing to know in terms of the step-by-step how these processes occur. Now, in this particular figure, we show the functional properties of myoglobin, and this is in the form of an oxygen binding curve. On the y-axis, we have the fraction of binding sites that are occupied by a ligand, and on the x-axis, we have the oxygen concentration, which is expressed in terms of the partial pressure of oxygen, which is generally given in terms of millimeters of mercury, or which is also the same as torr. Now, we see that in all these curves here that they have a hyperbolic shape. The P50 is what we generally use for the measure of oxygen affinity. So the P50 is defined as the oxygen concentration when half the binding sites are occupied. And therefore, half of the binding sites in this particular case would be 0.5. We see that the red curve pointed by the red arrow is oxygen binding at 40 degrees centigrade. And we can see that is right shifted relative to the curve pointed by the green arrow, which is the oxygen binding at 10 degrees centigrade. So immediately we can tell that the P50 is dependent upon the temperature and decreases as the temperature increases. So that's how the P50 can be used very effectively to understand the oxygen binding. To be a little bit more quantitative, if we look at the red arrow and go down to the oxygen concentration, we see that the P50 for this curve is right around 3, whereas the curve pointed by the green arrow has a P50 that is right around 0.2. So again, these give you a pretty good measure, an idea of how to interpret an oxygen binding curve. Now, human hemoglobin is different from myoglobin. Myoglobin is a monomeric protein. Human hemoglobin is a tetrameric protein formed from 2 alpha chains, which is shown in red in this figure, and 2 beta chains, which is shown in blue. You have alpha 1, beta 1 interfaces that are the alpha and betas along the vertical axis, each vertical one. And then you have an alpha 1, beta 2 interface as these 2 dimers interact with each other. So a better way of saying or describing the hemoglobin tetramer is to say that it's a dimer of dimers. Now, the functional properties of hemoglobin are significantly different from myoglobin. The first thing we can tell from this particular figure here is we look at the P50s, the myoglobin curve has a P50 right around 1 torr, whereas the P50 for human hemoglobin is right around 25 torr. So it's obviously that human hemoglobin has a lower oxygen affinity than myoglobin. The other noticeable feature is that myoglobin has more of a hyperbolic shape, whereas hemoglobin has a more S-shaped or sigmoidal shape. This is related to its function. This means that the binding of oxygen to the first subunits increases the oxygen affinity of the remaining subunits within the assemblage. And the reverse is also true. The loss of ligand from the first subunits decreases the oxygen affinity of the remaining subunits within the assemblage. Why is this important? Well, what we show here is the red to the right colored region is the oxygen concentration that's present in the human lungs, whereas the purple is the oxygen concentration that's present in the capillary bed. And we can see that in the lungs, human hemoglobin has virtually all of its binding sites occupied, whereas in the tissues, it has about half the binding sites occupied. So in approximately every round of circulation, the human hemoglobin tetramer is able to deliver about half its cargo load, or 2 molecules of oxygen per cycle. Myoglobin, on the other hand, would not be able to deliver any oxygen because it would be nearly 100% saturated at 75 torr versus 30 torr. So we're going to talk about right now the major mechanism of allostery, and that is the cooperative oxygen binding of hemoglobin. And as I stated before, cooperative oxygen binding is the binding of oxygen to one subunit, increases the oxygen affinity of the remaining subunits within the assemblage. Note that oxygen is the ligand that is the allosteric effector, and it's also being affected, depending on which site we're talking about. This means the allostery is homotropic. And model of homotropic allostery, or rather cooperative oxygen binding, is given by the MWC model. In this particular model, it's presumed that there's 2 energetic states, a low oxygen affinity T state, which is shown as a square, and a higher oxygen affinity R state, which is shown in a circle. In the absence of ligands, which is blue, the structure tends to be mostly T. The population tends to be mostly T state and not R state. However, the binding of oxygen, which is shown in red, shifts the equilibrium stepwise so that you'll have more and more R state population that's present. And as the R state population becomes more populated, you have higher oxygen affinity that's being observed. That gives us that S-shaped curve that we've measured experimentally. Now, the beauty of this model is that we can make one generalized rule that will help you throughout the rest of the phase. And that is anything that preferentially binds one state will stabilize that state and shift the equilibrium toward that direction. So, for example, molecules that preferentially bind the T state will lower the oxygen affinity of the hemoglobin. Examples of this are protons, 2,3-BPG, and carbon dioxide. Conversely, anything that preferentially binds the R state is going to increase the oxygen affinity of the hemoglobin. In this particular case, small molecules that bind the heme group, like carbon monoxide, nitric oxide, and molecular oxygen, will preferentially stabilize the R state. Now, structurally, the R and T states can be, at least to the first approximation, be understood in terms of two quaternary structures. We have a high affinity R state structure, which is shown with the red heme groups, and we have a lower oxygen affinity T state structure, which is shown by the magenta groups. And these two structures are very similar, but they differ in their quaternary structure by about a 15-degree rotation of one alpha-beta pair relative to the other. This is how we define an R state and a T state to the first approximation. And again, this is because you have one interface that's very, very tight and one interface that's very, very loose. And an important point is that the loose interface includes the F helix. And if you recall, the F helix is attached to the heme group. So somehow this quaternary structure change is tied to the oxygen affinity of the heme group through the F helix. Now, this is I'm going to go through some details to explain why this occurs. You don't necessarily need to know the exact mechanism, but you do need to know that the R state has a higher oxygen affinity than a T state. So in this particular figure, we have the heme group that's bound to a ligand, in this case carbon monoxide, and it's initially, the heme iron, which is shown in magenta, is initially within the heme plane. However, as soon as the ligand is released, it docks into a site that's within the distal pocket, and immediately the heme iron moves out of the heme plane. As you see the heme iron moving out of the heme plane, you see the F helix being pushed down and away from the heme plane. And as the proximity is being pushed down and out of the heme plane, so is the F helix. And that motion is going to be transmitted into the interface. And that's what we see here. This is the deoxy structure. This is the R state structure. So as you're going from one to the other, you see the F helix rotate just a little bit. This rotation changes the interface that occurs. And as one side of the interface occurs, the other side of the interface has to adjust to compensate. This means that particular heme group is going to be rotated as well. This means as one oxygen, as the ligand state changes at one subunit, it's going to impact the F helix of the neighboring subunits within the assemblage. And this gives rise to the cooperative oxygen binding. So the cooperative oxygen binding within human hemoglobin is transmitted through the F helix. Now, there's also heterotropic allostery within human hemoglobin. So, for example, the binding of something like 2,3-BPG or bisphosphoglycerate is going to alter the oxygen affinity of human hemoglobin. And this particular mode of allostery is referred to as heterotropic allostery because 2,3-BPG is a different ligand than oxygen. So formal definition of heterotropic alteration modulation of oxygen affinity at one site on a protein is influenced by the binding of a different ligand at a different site on the protein molecule. And this particular figure gives you sort of an explanation of 2,3-BPG or bisphosphoglycerate. The binding to the left, we have an oxygen binding curve, which shows percent saturation of oxygen on the y-axis and the partial pressure of oxygen measured in millimeters mercury. We see that as the 2,3-BPG concentration increases, we get a shift in the oxygen binding curve to the right. That means the P50 is getting larger and the oxygen affinity is getting smaller. To the right, we show where the crystallographic evidence is where the 2,3-BPG binds. It binds in a highly charged, positively charged pocket formed between the beta-1 and beta-2 subunits. In the figure to the right, we show the surface of the hemoglobin molecule, which is shown in white, with the areas which are negatively charged shown in red, and the areas which are positively charged shown in blue. And the 2,3-BPG binds into the positively charged pocket, which is indicated by the arrow. Now, this is really not that surprising because 2,3-BPG or 2,3-bisphosphoglycerate, you have the glycerate, which is a carboxylic acid, which is negatively charged. You have two phosphate groups, which carries negative charge. So you have a lot of negative charge that's binding to this very positively charged pocket. In this figure, we have 2,3-BPG, which in this figure is labeled as DPG, bound within its binding pocket. Again, 2,3-BPG is negatively charged, and it interacts with a bunch of positively charged amino acids. In this case, we have histidine 2, lysine 82, and histidine 143 from one subunit, and the equivalent residues from the neighboring subunits. So one subunit is beta-1, and the other subunit is beta-2. Now, this explains why the 2,3-BPG binds to this particular part of hemoglobin. Why it preferentially binds to the hemoglobin is that the pocket is large enough for ligand binding to occur. When the R state's present, this pocket is shifted a little bit, and it's much smaller, and therefore the charged 2,3-BPG cannot bind to it. Therefore, 2,3-BPG preferentially binds the T state and lowers the oxygen affinity of hemoglobin. 2,3-BPG also plays a major role in why fetal hemoglobin has a higher oxygen affinity than hemoglobin A. And the reason for that is because it has a lower 2,3-BPG affinity. An explanation can be seen in the structure. If you compare equivalent residues, the histidine at position 2 is the same. The lysine at position 82 is the same. However, the positively charged histidine is replaced by a neutral, uncharged serine residue. It still has the ability to interact with the phosphate group, that is, it can form a hydrogen bond, but you lose the electrostatic interaction. So that's why hemoglobin F, or fetal hemoglobin, has a higher oxygen affinity than human hemoglobin or adult hemoglobin. Why is this important? It's because this allows the fetus, when it's growing, to extract molecular oxygen from the mother and therefore allows the fetus to grow. Without the ability to extract oxygen from the mother, the fetus would not be able to survive. One other issue that involves the 2,3-BPG binding pocket is that it's mainly dealing with charged molecules. So any molecule that has a lot of charge has the capability of binding to this pocket between the two beta subunits. This includes small molecules such as ATP, inositol pentaphosphate, and inositol hexaphosphate. And in some species, these are the actual physiological effectors of hemoglobin function. 2,3-BPG is made from 1,3-bisphosphoglycerate in a reaction as catalyzed by 2,3-BPG synthase. And 2,3-BPG is degraded by 2,3-bisphosphoglycerate phosphatase, producing 3-phosphoglycerate. Both 1,3-bisphosphoglycerate and 3-phosphoglycerate are intermediates of glycolysis. Now, the activities I described, the synthase activities and the phosphatase activities, are present at the same active site on the same enzyme, and this enzyme is called bisphosphoglycerate mutase, abbreviated BPGM. And the rate of synthase activity to phosphatase activity is controlled to some degree by the pH. Note that 1,3-bisphosphoglycerate and 3-phosphoglycerate are intermediates of glycolysis, so therefore the ultimate precursor for 2,3-BPG is glucose. Carbon dioxide and proton ions both are important heterotrophic effectors that lower the oxygen affinity of human hemoglobin by preferentially stabilizing the T state. Carbon dioxide can enter the red blood cell through the RHAB protein, and bicarbonate ions can enter the red blood cell through band 3, which is the anion antiporter. On both sides of the interface, carbon dioxide and bicarbonate can be interconverted by the reaction shown above. This reaction is catalyzed by carbonic anhydrase. Now, 4 molecules of carbon dioxide will react with the amino terminal groups of each subunit to form a carbonyl adduct. This particular adduct only occurs in the T state. So that is one way that carbon dioxide will bind, and it will preferentially stabilize the T state until the oxygen concentration increases and the carbon dioxide is forced off of the amino terminal groups and into solution, where it exits the cell and leaves the body through respiration. Now, protons can quickly traverse the membrane and will impact the oxygen affinity by lowering the oxygen affinity of human hemoglobin. This is referred to as the Bohr effect. The major Bohr group has been identified as histidine 146. However, there are additional Bohr groups, and this is known because histidine 146 has been mutated to a non-Bohr group of molecule such as a serine. This reduces the Bohr effect, that it still lowers the oxygen affinity, is still dependent upon the pH. However, the magnitude of dependence has been reduced, but it's still there. So that means there are additional Bohr groups. These Bohr groups could be several different amino acids, all of which have their pKa slightly adjusted by the 2 quaternary structures that are present in hemoglobin. Now, the candidates for the Bohr group are the same candidates for any particular amino acid that can act as a buffer. That is, they have to have a side chain that can exchange a proton. This limits the potential Bohr groups to histidines, cysteines, aspartic acids, glutamic acids, arginines, lysines, and tyrosines, or the propionic acid side groups of the heme. So all of these are potential Bohr groups. But the other important point that you need to remember is that hemoglobin also acts as a buffer within the red blood cell, and all the cysteines and all the histidines are most likely to play an important role in the buffering capacity because their pKa's are near physiological pH. Now, to summarize, according to the MWC model, anything that preferentially stabilizes the T state will lower the oxygen affinity, and any molecule that preferentially stabilizes the R state will increase the oxygen affinity. So small molecules such as protons, 2,3-BPG, and carbon dioxide will preferentially bind to the T state and will lower the oxygen affinity. Small molecules such as carbon monoxide, nitric oxide, and molecular oxygen will preferentially bind to the R state and increase oxygen affinity. Now, carbon monoxide and nitric oxide is also more complicated than that because in addition to preferentially stabilizing the R state, they are competitive inhibitors of oxygen bonding. So at high concentrations of CO, oxygen will not bind, and you get carbon monoxide poisoning. And that is the end of this particular talk.